Hello, everyone. My name is Mitchell Connon. I am a third-year biomedical student working in the microdevice laboratory with Dr. Smitha Rao. Today, I'll be presenting my summer undergraduate research fellowship, Durotactic Migration of Cancer Cells on Engineered Scaffolds. In order for cells to grow, they need some support. In a lab, typically cells are grown in culture plates. Research has shown that the stiffness of the material used affects the response of the cell. For example, cancer cells prefer a stiffer substrate. Through my work in the biomedical microfluidics lab, I am exploring the use of electrospun, three-dimensional nanofiber scaffolds on different mechanical stiffnesses and how they affect different cells. Thus, the work is based on evidence that migration of cells in diseases such as cancers is impacted by substrate stiffness, a type of directed migration called durotaxis. For my experiments, I've used electrospun nanofiber scaffolds made of polycaprolactone, or PCL for short. By changing the parameters of fabrication, the stiffness of the scaffolds produced were altered in a controllable manner. I used two distinct scaffolds and compared them against the standard tissue culture protocol. The cells I used were renal cancer cells 7860, MCF7, and 184B5, which is a normal breast tissue cell. These cells were seated on aligned scaffolds that were spun at different speeds, 275 and 250 RPM, to obtain a different stiffness. On these scaffolds, I characterized the cell's response through cell viability, expression of biomarkers, and optical imaging. All experiments were carried out using standard tissue culture protocol with 3 repeats. The data indicated CK18 proteins could possibly upregulate for cells 184B5 and MCF7. Looking at the graphs for MCF7, 7860, and 184B5, these cell lines are shown to prefer a softer substrate over a more stiffer one. Based on our work, it is clear that further investigation of the nanostructures and the microenvironment impacting cancer growth needs to be investigated because the major effect it has on cell communication, proliferation rate, and general health. In conclusion, mechanical stiffnesses affect cell growth and migration. Detailed analysis of the cellular response could provide clues regarding cancer migration and metastasis. Future work will include Western blotting to identify regulation of cancer-specific biomarkers and quantitative analysis using flow cytometer. I would like to acknowledge the support from the Pavlos Honors College and Portage Health Foundation Summer Undergraduate Research Fellowship. Thank you.